We've been working in two areas, one that's somewhat come to an end, but it was very important clinically. And this had to do with the molecule hepatocyte growth factor that works with the c-Met receptor. And c-Met is an oncogene, and it is directly involved in metastasis of cancer. And we, in collaboration with biologists, studied the structural biology of this, and using the fundamental information we obtained structurally, we were able to engineer a variant of this molecule, it's a protein molecule, to make an antagonist that actually was clinically responsible in animals, not in humans yet, but in animals, and actually was broadened to also look at the vascular endothelial growth factor, or VEGF system, and so we generalized a principle of intervening with heparin-based drugs. Another area that's particularly of interest to us is the proteasome degradation pathway that regulates the level of proteins in all human cells, and of course that's part of apoptosis and cell death. And our line of research is really to understand the mechanisms of the proteins that drive this process so they can be used therapeutically to manipulate or modulate that process. What we hope from the basic knowledge that we generate is that it will both drive new biology, people to try new things, to test the hypotheses and the theories that we put out about function. So it's really about structure and function relationships. So both in the basic research with my molecular biology colleagues, but then leading further to actually develop agents that will interfere either to enhance or to inhibit key cellular processes. That's really the underlying point of trying to impact medicine. We primarily use nuclear magnetic resonance, or NMR, but we also use a variety of other tools, solution X-ray scattering, various biophysical tools to look at binding and complexes. In the NMR field, which is really our specialty, the biggest key element has always been sensitivity. NMR is a very powerful tool and can lead to all kinds of information about structure and dynamics. And usually we find in human and mammalian proteins is that we're stuck fighting sensitivity all the time. Given sensitivity, spectroscopists around the world and our group can devise all kinds of new things, but sensitivity seems to be the holy grail.